All right, so this is our second part of enzymes for honors. You should have already gone through kind of interpreting enzyme graphs and looking at basic properties of enzymes. This is going to take us through how enzymes work and some factors that can impact how those enzymes work. Well, how enzymes work. So we have this molecule called the substrate, and the substrate is essentially the reactant in a reaction when an enzyme is working on it. And if we look at the diagram that we have in our right-hand corner right here, we see this kind of pinkish color right here. This is our enzyme, and it's got this curved surface on it right here called the active site. And if we look up here, this is our substrate. So this is the molecule the enzyme is going to work on, and we notice that it's got a matching physical structure right here. So what's going to happen is inside all living things, substrates are going to physically bind to enzyme at these active sites. And these two perfect pieces are going to come together just like puzzle pieces and form what's known as the enzyme substrate complex. Now, while you have this enzyme substrate complex formed, what's actually happening is that the molecule itself, so this enzyme is actually physically positioning the substrate so that it's going to be easier for it to reach its transition state. What that means. The transition state, remember, is when you've broken apart all of the atoms inside of a substrate or inside of a reactant, and you're getting ready to reform them into product. And we said you have to add a bunch of this activation energy in there in order to get these substrates to that transition state. By physically positioning the molecule at the active site, what the enzyme does is it makes it easier and more stable for these substrates to reach that transition state. And so it lowers that activation energy and allows that progression from reactant to product to occur a lot more quickly. So our last bullet in kind of the big picture, so this means that enzyme-mediated or enzyme-controlled reactions can occur more quickly and require less activation energy. So remember, we're not changing the substrate or your reactant. We're not changing the product. We're just making that conversion between the two happen a lot more quickly. And again, it's through the physical positioning of the molecule at this active site. So this is why, if we kind of remember, I told you guys that shape of your proteins is going to be hugely important. So when we looked at that active site on the last slide, remember this active site had to be this perfect shape right here for the substrate to just click right in. When proteins bend and fold, so from primary to secondary, tertiary, and maybe quaternary structure, depending on the protein, that folding and bending is going to form the active site of the enzyme. If you alter the amino acid sequence, or if you change, remove amino acids, if you do anything, what can happen is when that folding and bending occurs, you might not get the perfect shaped active site. And if you change the shape of the active site in any way, shape, or form, well, then the substrate can't bind to it anymore. And therefore, that enzyme becomes useless, and those reactions are going to start to happen a lot more slowly. So this folding, bending, the sequence of amino acids is hugely important for your enzymes to be able to do their jobs. So kind of putting that all together, protein folding forms the active site, and this is where your substrate actually binds to. If we have mutations or changes inside of our DNA, DNA is actually going to contain the code for how we make or build our enzymes inside of our cells. So if we change that code, what we can do is start to produce enzymes that are the wrong shape. And cells that have these mutations or altered DNA codes are going to have some altered function. So they might not be able to actually have some reactions occur at the rate they should be in order to sustain life because of this mutation. We're going to talk about three things. So you should be on part 4 right now that impact the shape of an active site. So it can impact enzyme function. So there's mutation. So this is when you have a change in the DNA, makes a different amino acid sequence. Protein doesn't fold the right way. If enzymes don't fold the right way, because they're proteins, you get the wrong shaped active site, and it can't do its job. We also have pH, high temperatures, and allosteric inhibitors. Low temperature was in your notes, but low temperature doesn't actually change the shape, but it can impact function. So let's start at pH. So you should have already filled in 1 for mutation. So now we're on pH and temperature. So enzymes have this optimal or best temperature and pH they can function at. When you move outside of these optimal conditions, what happens is the enzyme will start to change shape. We call this process of an enzyme changing shape due to high temperatures or altered pH denaturing. Denaturing is just the technical term for an enzyme changing its shape because of these things. And we remember shape, hugely important. Substrate needs to fit into that active site. I mess with the shape. I mess with the active site. It can't bind anymore. So denaturing occurs because of those interactions between the ion groups. So what happens when you change pH? Let's see if this is going to be nice. And actually, oh, it's not nice. There's an animation on this slide that doesn't seem to be working. What happens when you actually change the pH is, remember that acids produce a lot of H plus or positive ions, and bases produce a lot of OH minuses or negative ions. When I put something, a protein, in a very acidic environment, it's in a very positive environment. So what can happen is those R or variable groups can start to interact with all of those positive charges around it, and it'll change its shape. If I take a protein or an enzyme rather and put it in a very basic environment, well, then it'll start to interact with all those negative ions that are inside the environment, and it can change its shape as well. That's literally the chemistry behind why pH matters. Remember, acids, because they're making more H plus ions, are more positive. Bases, because they're making more OH minuses, are more negative. In either case, you're causing some altered sort of bonding to occur between those R groups, and you're changing the shape of that protein. You also have this entire group of chemicals called inhibitors. So biologically, sometimes we don't necessarily want enzymes to be working as quickly as they can be. So we have a whole class of molecules, these are proteins as well, that inhibit or reduce the function of our enzymes. Competitive inhibitors are going to be inhibitors that compete for the active site. So we've got an image up here, and it's showing this blue structure as a competitive inhibitor. So if you notice, it's got a part of its molecule that's perfectly shaped for the active site. If it gets there first, it'll bind to that active site and it won't let go. So now if a substrate or a molecule that that enzyme should be working on comes over, no place for it to fit in. So now that reaction slows down. There are also non-competitive or allosteric inhibitors. What's going to happen is some enzymes contain what's known as an allosteric site. So it's a second location that's formed on the molecule when that protein bends. So this right here, if we look up at the diagram, this V shape is the active site. And this second little kind of notched in area right here is the allosteric site. Allosteric inhibitors are also called non-competitive because they're not trying to get to the active site. They're not trying to beat the substrate there. What they do is they go to the secondary site, and when they bind there, they actually slightly alter the chemical properties of the amino acids and cause that active site to change shape just enough that now, so if you look over here, now it's a little squared instead of being a triangle. So if we look at my substrate's not going to fit anymore in that active site because this allosteric inhibitor has altered that active site so it doesn't fit. So inhibitors can also reduce enzyme function. Competitive inhibitors compete for the active site, and allosteric or non-competitive inhibitors bond to an allosteric site, which is a secondary location on the enzyme, change the site of the active, change the shape of the active site so substrate can't bind. If we talk briefly about low temperatures, so we said at high temperatures, your protein's going to denature. It's going to actually change shape. But at low temperatures, your enzyme is actually not changing shape. If we kind of look right here, we've got a phase change graph, and it shows us very briefly what's happening in terms of molecular behavior at each level. As we go kind of in decreasing, so remember, as molecules get colder, they move or vibrate less. And so they're actually moving around less, and the opposite. So if I were to increase temperature, my molecules are moving more. In order for an enzyme to work, it needs to actually bump into or come in contact to a substrate, which means the molecules need to be moving around. As you reduce temperature lower and lower and lower, eventually you can get to a point where the substrate and enzyme literally aren't moving enough that they come into contact with each other. This is what happens at low temperatures. It is not that the enzyme is denatured. It is that there is not enough kinetic or movement energy to allow the substrate and the enzyme to come together in order to move the reaction along.